It's a great pleasure to be here. It's a wonderful experience of a lifetime to be a Radcliffe Fellow. I'd like to thank Dean Liz Cohen. I'd like to thank Associate Dean Judy Vishniak, all the staff at the Radcliffe for the wonderful work that they do to keep us safe and happy through the year. I'd like to thank my fellow fellows that have also given me such a good ride through this year. And I'd like to thank the people with whom I've been working this year, in particular the lab of Andy Knoll in Earth and Planetary Sciences, Andy himself and his lab, in particular Elizabeth Siebert, who's a Harvard Fellow, who's been working with me during this year, teaching me about new things, and also my Radcliffe research partner, Hannah Byrne, who's been working together with a postdoc in my lab in Paris to move forward with some of our work. So there's many people I'd like to thank. Also Dan Needleman in molecular cell biology here, who's been a great inspiration to me too. So with that, let's dive into the ocean. So I'd like us to think about the ocean for the next hour or so. I'd like to think about our planet ocean. I think we all appreciate that the ocean occupies more than 70% of the surface of the Earth. And we look at it, and I think we all appreciate that it's huge, that it's awesome, that it's powerful, right? But when we think about, how does it connect to us? How does it connect to our daily lives? It's maybe a bit more abstract to bring the ocean into that intimacy with ourselves. So let's go back to the very beginning. Let's go back to the stromatolites, the fossilized remains of the very first cyanobacteria, as far as we know. And it's within these organisms that this incredible machinery called photosynthesis first evolved. In my opinion, this is perhaps the greatest evolution, the greatest invention of evolution that we know about, that evolved very early on and performs incredibly important things. If we think about it for a second, what photosynthesis does is it converts light energy into chemical energy, okay? That's what we try to do when we generate energy out of solar energy. We're not very good at it yet, we're getting better. But plants and photosynthetic organisms do this incredibly well by generating chemical energy that can be utilized in different ways from sunlight, from the energy in sunlight. Photosynthesis also converts carbon dioxide in the atmosphere into organic materials. So everything, including ourselves, we are all built from this organic materials, that we are literally what we eat, or all of that organic material comes ultimately from primary production generated through photosynthesis by the conversion of carbon dioxide into organic materials. And then photosynthesis splits water to generate oxygen, which of course we and many of our co-inhabitants of our planet also breathe. So it was a remarkable invention of evolution that really changed the history of our planet and permitted life on Earth to really evolve as to how we know it today. Here we see the evolutionary record of life on Earth beginning in the Archean, sort of 3.5 billion years or so ago, where we have the first evolution of oxygenic photosynthesis within these cyanobacteria that formed ultimately these stromatolites. And this started generating whiffs of oxygen, so there was no oxygen on Earth before life first evolved, right? And then finally these whiffs of oxygen began to be generated, ultimately leading to what we know as the Great Oxidation Event. And then slowly, as oxygen began to stabilize, still at pretty low levels, not yet at today's levels, then we see the evolution of different forms of life that were very likely stimulated by the photosynthetic processes. So the first reactions that we know of are coming from organisms such as the cyanobacteria, which later on, following the increase in oxygen, gave rise to more complex eukaryotic cells, which are cells like our own cells, which contain DNA packaged inside the nucleus, which have mitochondria to generate energy inside them, and so forth. So this was the great first innovation that was likely permitted by evolution, by photosynthesis in the first place. And then as these eukaryotes continued to evolve, they were still single-celled, okay? They ultimately gave rise to photosynthetic eukaryotes. These eukaryotic cells had learned how to do photosynthesis by acquiring cyanobacteria and incorporating cyanobacteria inside those cells. And then following the evolution of these first photosynthetic eukaryotes, we likely had another bump in oxygen up to pretty much modern-day levels of around 20%, which then permitted the evolution of multicellular life, the evolution of more complex life forms, most notably in the Cambrian explosion, where all kinds of weird and wonderful multicellular organisms first appeared in the fossil record. And then we have a third moment when photosynthesis appears to be particularly important, which was the evolution of land plants, so multicellular organisms that can photosynthesize, beginning to colonize the land. We had another bump of oxygen going up to sort of 30% here, and this likely permitted the evolution of giant insects, of giant animals up to dinosaurs and so on. And then these levels finally went back down again to modern-day levels of about 20%. Fortunately, otherwise we may be living with these giant insects and animals even today. So here we see the history of life on Earth that was very much permitted by photosynthesis. And very much enabled by different evolutionary events that happened in the ocean. So life evolved in the ocean, we're pretty certain of that. And through different aspects, we now know that the ocean very much sustains life on our planet for all kinds of different organisms. And in our own particular way, for ourselves, the ocean protects us. It protects us by feeding us, providing us with fish and so on. It protects us by absorbing excess heat, by absorbing carbon dioxide in the atmosphere. Without the ocean, the temperature of our planet would be even higher than it is today. And the ocean also generates oxygen through these photosynthetic organisms that are present. The ocean, finally, is also a resource in many different ways, not only for tourism, also for desalination, extraction of minerals, all kinds of different resources that we know are important. And we sort of tend to think that the ocean is so big, it can sort of absorb whatever we throw at it, right? But in fact, that's not true. If you take all of the water that is in the ocean and you bring it all together into one single drop, sitting on top of a dry Earth, you begin to see how little water we actually have. And the majority of this is ocean water, okay? And it's really very, very little. If we look at fresh water, it's a little drop of water sitting on top of the ocean water, of the salt water that you probably can't see at the back. So the ocean is really not as big as we think. We're throwing all kinds of insults at it, but it can only withstand a certain amount. And we know today that the ocean is very much under pressure. It's under pressure in several different ways. On the one hand, through the increase in temperature in the atmosphere, due to greenhouse gases that are present in the atmosphere. are causing increases in the temperature of the ocean. The increase in atmospheric carbon dioxide is mixing with water, and that simple reaction of carbon dioxide plus water produces carbonate, produces bicarbonate, which are acids, and so the oceans are slowly acidifying in a pretty dramatic way. And then through phenomena that we don't really understand, the oceans are also losing their oxygen. There is a process of deoxygenation that has been observed in many locations around the world. So this is a sort of third insult that is happening to the ocean collectively. And in a global context, we can see the increase of the increase of sea levels caused by the melting of the glaciers that are affecting coastal ecosystems. We have effects on coral reefs caused by acidification of the ocean, and so on. So the ocean is very much feeling the pressure of humanity today. So it's in this context that we really launched this expedition called Terra Oceans back in 2009. And what we wanted to do here was to sort of get an idea of the state of the ocean at the beginning of the 21st century, because we never really, nobody has ever done this before. We don't really have a zero time point for the state of the ocean. So we set out to do this project way back in 2009. And we specifically wanted to study the state of marine ecosystems, in particular the microscopic plankton that I'll introduce you to in a second. And we were very much inspired by previous expeditions as well, learning from the past to try to move forward, standing on the shoulders of giants, people who had done this sort of thing before. And here are a couple of examples of previous expeditions that I'm sure you have heard of. Certainly this one on the HMS Beagle from 1831 to 1836. There were two particular gentlemen on board here. Captain Robert Fitzroy, he was the captain of the ship, and his job was to map out the coastlines of the different countries that they traveled around. This is South America. You see, he did a fantastic job doing this work, on this expedition. But he was also very much a scientist in his own right. In those days, scientists didn't exist. They were called natural philosophers. And I think that describes very much who these kinds of people were. And in the case of Fitzroy, he went on to develop what we now call the storm barometer, which is a remarkable invention to allow us to predict the weather, right, based on the atmospheric pressure, to know when it's going to rain or when it's going to be fine. So he developed that during his lifetime. And also on board this expedition, there was the young Charles Darwin. And I'm sure he needs no introduction as to what he achieved in his lifetime in providing us with a theory for the origin of species and mechanisms of evolution and so on. And then there's a second example that I want to talk about briefly, which is the HMS Challenger expedition. This was performed about 50 years later, 1872 to 76. This was performed by Thompson and Murray, and they went out over a 5-year expedition. You can see where they went here, to basically test the idea that was prevalent at that time, that there was no life in the ocean below the surface layers, below where the light can penetrate. It was believed at that time that the ocean was azoic, devoid of life. So they went out on this expedition, they dredged and pulled up all kinds of incredible organisms to show that, yes, indeed, there is life at these depths of the ocean. And many of these samples were subsequently taken by this guy called Ernst Haeckel, who was a German. He was really very much a natural philosopher in the sense that he was sort of half scientist and half artist. And he generated these incredible images of these microscopic organisms that he was observing in his very primitive microscope, that ultimately gave rise to the movement of Art Nouveau, very much inspired this whole idea of living forms being used to create new architectural forms. This is the World Expo in Paris in, I think, 1902. You can see the Eiffel Tower back here. This doesn't exist anymore, sadly, but you can see how it was very much inspired by these microscopic life forms that were drawn by Haeckel. Haeckel was also the father of ecology. He coined the term ecology, which has now become, obviously, a formal science in itself. And this movement of architecture of Art Nouveau subsequently continues today through, organic architecture, which, we see many examples around. So we see how life continues to inspire beyond the science itself of studying life on Earth. So these were the sorts of inspirations that we got from our project for initiating the Terra Oceans project. And what we specifically wanted to study in this project were the planktonic organisms, which we can consider as sort of the invisible multitude. When you think about life in the ocean, I'm sure you don't think about plankton, you think about sharks and whales and so on. But in fact, the plankton, these microscopic organisms that drift with the currents, really do represent the majority of life in the ocean. They're at least 50% of the biomass of life in the ocean, probably considerably much more than that. And they consist of all kinds of different organisms of different shapes and sizes that you can see in this mandala that I'll introduce you to in a second. And these organisms have all kinds of very important functions. They are the base of the oceanic food chains that lead up to the fish and the larger organisms. The photosynthetic component of the plankton, just like plants on land, they sequester carbon dioxide out of the atmosphere and they bring it into organic materials in the ocean. Through photosynthesis, they also generate the oxygen that is so important for us and many other organisms. And because the ocean is deep, some of these organisms, when they die, they will sink to the bottom of the ocean. And they'll take the carbon that they have produced, the organic carbon, they'll take it with them all the way to the bottom of the ocean, effectively taking carbon out of the carbon cycle on the surface of the Earth, sequestering it for geological periods of time that can ultimately form the fossil fuels that we're so avidly burning today. And we know through studies from many people around the world, including also many eminent scientists in Harvard, that plankton, on the one hand, can affect climate. They've affected climate of the Earth during the history of life on Earth in several different ways. And we know also that these organisms are affected by climate change. So there's a kind of feedback between the two, between climate and planktonic organisms, such that plankton are really at the very heart of the climate machine that regulates the function, that regulates the climate system of the planet. So, what's in the plankton then? You can see this mandala here of nice little organisms, but let's go and dig a little deeper and let me introduce you to the different kinds of organisms that are present. I'll start with these guys that we call the protists. These are cells that consist of eukaryotic features, just like our own cells. The difference is that these guys are single-celled. Typically, they exist as single cells. Sometimes they will form chains, as you see here. And you see they have different kinds of shapes and sizes. This is about, each one of these cells is between 1/100th and 1/10th of a millimeter in size, I would say. Some of them have structures like little pillboxes, like you see here. Some of them look like little spaceships, all kinds of weird and wonderful shapes. Some of these are photosynthetic. Some of them parasitize other organisms. Some of them eat organic material like we do. And if we add all of these up together in an average liter of seawater, we can find up to 10 million of these organisms in that volume of water. Okay. So these are the protists. On top of the protists, we have what we call the zooplankton. These are different kinds again, different kinds of shapes and sizes. They generally tend to be bigger. And these are the grazers in the ocean. These are the cows and the sheep, okay, of the oceans that go around eating typically those protist organisms that I showed you in the previous slide. And these can be present at up to 100 individuals in an average liter of seawater. And then getting smaller, we've got the prokaryotic organisms, the bacteria, the archaea, which can be present at up to 1 billion cells in an average liter of seawater. Some of these are photosynthetic. Some of them are related to those initial cyanobacteria that gave rise to the origins of oxygen on Earth. And then even smaller than the bacteria and archaea, we have the viruses and the giant viruses, which again are all kinds of different weird shapes. And these can be present at up to 10 billion individuals per liter of seawater. These are not pathogenic for us, but the role of these guys is to infect the other components of the plankton, okay, to essentially ensure that there is equilibrium in the ecosystems. If one organism tries to take off, tries to go too strongly, a virus will show up to cut it down, to ensure that equilibrium is maintained. So I sort of consider these organisms as like the lubricators of the ecosystem to ensure that excesses are not tolerated and to ensure that evolution moves forward as organisms adapt. So we have all of these organisms that are present in ocean water, and they're present in different amounts, and they're also present very much in different sizes. What you see here is a video of a zooplankton, the large shrimp-like organism called a diatom, called a copepod, with a diatom sitting on top of it, this round cell here, which is one of the protists. That's the diatom here. And then sitting on top of the diatom is a bacterial cell here, okay? So we're going through several orders of magnitude here, equivalent to going from an ant to a brontosaurus, if that maybe means more to you. And we don't even have the virus. The virus would be sitting on top of that bacteria, okay? So these were all of the organisms that we wanted to study in the Terra Oceans expedition. It had never been done sort of from end to end before, so it was a challenge to put together protocols to sample all of those organisms. And we also wanted to do this on a worldwide scale because the ocean is one. It's a single ecosystem, but to understand that ecosystem, you have to sample it globally, okay? So we performed this 4-year expedition that is summarized on this slide here, where you see in 2009 we left France, we went through the Mediterranean, through the Red Sea, down the Indian Ocean, across the South Atlantic, into the Southern Ocean in Antarctica, and then went out the South Pacific towards Tahiti, up to Hawaii, back to San Diego, through the Panama Canal, up to New York and back to France over 3 years. And then in 2013, we performed a second expedition going around the Arctic Circle up here because the Terra sailboat is able to withstand the ice, able to withstand the pressures of the ice, and is able to be able to do that sort of extreme expedition quite aptly. So during this summer season here, it took us 6 months to get around first the Northeast Passage above Russia, and then through the Northwest Passage above Canada. Many explorers have lost their lives looking for those passages in the past, right? But these days it's possible to get through because of the melting ice up in the Arctic. But Terra is anyhow the first sailboat to have gotten through both northeast and northwest passages through in one season. So during this whole campaign, we sampled at all these different sampling points that you see here marked out on the map. Our objective was really to do the same thing wherever we went, so that ultimately we could compare, samples that we got in Antarctica with samples that we got in the Arctic, for example. So there's a lot of mundane work to really generate high-quality samples that would allow us to subsequently compare sort of everything with everything. So at the end of this whole adventure, we had sampled at 210 different sampling sites around the global ocean, and we'd brought back 40,000 samples to the labs involved in the Terra Oceans project for subsequent analysis. We brought back other data sets, other samples, which were also useful for some of the analyses we wanted to do afterwards. And then, so that's when the real hard work begins, to dig through these 40,000 samples and try to make some sense out of them. And we were very happy that we could that we were able to describe the first foundational results from the project in a special issue of Science that was published almost 2 years ago now, on May 22nd, 2015. And in this special issue of Science, we were able to publish 5 papers back-to-back describing the foundational results from the project. We got the editorial, we got the front cover, we got a lot of coverage in the scientific press and also in the daily press. We got also the front page of the New York Times. This is their view of plankton, not exactly microscopic, but at least it serves to get the message across. So we're very happy to create this splash of media coverage finally about plankton. And just let me show you briefly what is contained in some of those papers in Science. In one of those papers, we described what we call the ocean microbiome. So you've maybe heard of the human microbiome, the microbes that live on human beings. In this paper, we described the equivalent for the ocean, the microbes that live in the ocean, in particular the bacteria. We described this microbiome from 68 sampling sites within our data set, so we had pretty good global coverage of the global ocean. And within this microbiome, we described 40 million different genes, okay? That is close to saturation, so this is pretty much all of the genes that are present in bacteria in the ocean, at least in the top 100 meters or so of the water column. So it's a huge resource that is now publicly available that is being used by scientists worldwide. It's about 30 times. This is about 30,000 times more sequence than was generated by a certain Craig Venter in a similar expedition performed several years before us. And of these 40 million genes, you can imagine most of them are new to science. More than 80% of them were discovered on the Terra Oceans expedition, and the vast majority of them, we don't know what their functions are. So there's a lot of discovery to be done to try to understand what those functions are. So that was one paper. In another paper, we described the ocean virome, the collection of viruses that we could find in the ocean by methods that I won't go into. But in this paper, we described 5,500 different kinds of viruses in the global ocean. Prior to Terra Oceans, only 39 were previously known. So that gives you, again, an idea of the incredible rate of discovery coming from the project. And then for these protists, the unicellular eukaryotes, this gets a bit more complicated because we have an incredible diversity of organisms within the eukaryotic tree of life that is shown here, for many of which we have no cultured representatives. We don't know what they look like. And there's an incredible unexplored diversity that we know very little about. To give you an idea, within this tree of life here, we are sitting here within the animals, okay? And the closest branch to us are the sponges. So here we've got animals and sponges, and then beyond animals and sponges, you have all this incredible diversity of organisms that we know very little about. So from this survey of these protists, what we described in one of these papers was about 130,000 genetic types. Let's not call them species, but sort of species. 130,000 different kinds of eukaryotes in the ocean. Again, this is saturating, so we've more or less found everything that is there. So if you give me another sample of seawater, we'll more or less just find what we've already found from this project here. And again, there's an incredible novelty represented in this data set. This represents more than 10 times the number of formally described species of marine eukaryotic plankton, so 90% are new, and about a third of them, we cannot assign them to any known taxonomic group within this tree of life. Okay, so whether these organisms represent very primitive groups, sort of at the center of eukaryotic diversity, remains to be seen. But there's an incredible amount of diversity that was revealed here. Now, all of this was done by DNA sequencing, very, very high throughput DNA sequencing, which gives you a one-dimensional string of G's, A's, T's, and C's, right? It doesn't really necessarily tell you what these organisms are. So it's very, very important to try to associate some sort of morphology, some sort of shape, some sort of forms with those DNA sequences. And so that is why we developed additional protocols to perform automated, high-content imaging of these organisms sitting on specialized microscopes we call confocal microscopes, such that we can get these very, very detailed views of what individual kinds of planktonic organisms look like. Here are a couple of examples in more detail. So using these different kinds of fluorescent dyes, we can look at the exoskeletons of these organisms. We can look inside these cells as well. This is a dinoflagellate inside. The red is a chloroplast. The green are membranes sitting inside the cell. This is about 1/10 of a millimeter, about 100th of a millimeter in size. This organism is a diatom, which has a glass silica cell wall. You can see how elaborate that is in light blue here. Going inside the cell, we can see the chloroplasts in red. The DNA is sitting inside the nucleus here in blue, in this very elongated vesicle here. So we have all kinds of shapes of these organisms. We have more than 1 million of these kinds of images, and we're now trying to associate the DNA sequences with these morphological information to try to figure out who is who. That's obviously a work in progress that is going to take some time. So now that I've sort of described to you what we know about the bacterial components, what we know about the virus components, what we know about the protist components in the ocean, we can take all of that data, we can throw it all together. We have that information from 68 different locations around the ocean. We can throw all of that data together and we can ask, well, who lives with who? Who is found in the same location as the other organisms, or who is never found with other organisms? So through this very large compute, we can define what we call in technical terms the interactome of the plankton ecosystems, which you can also consider as a social network of the ocean, the ocean's Facebook. So this took several months to compute. It's not particularly nice how it looks in the end, but this is the plankton interactome here. The different dots are different kinds of organisms, and the lines between organisms are either associations in green, so organisms that co-occur, that live in the same place, or in red, organisms that tend to exclude other organisms. Okay, so you can see some patterns in here. You can see some kinds of plankton that are sort of universally excluding other organisms, sort of universally antisocial. We can consider these as the Putins, the Donald Trumps of the plankton world that are very, very strongly negative. We've also got these very positive guys that are universally sort of green. These would be the Tom Brady's, at least in isolated communities, the Beyoncés, the Lady Gagas, and so on. So we have all these kinds of different plankton which we can tease apart and try to understand more about what they're actually doing. The sorts of conclusions that come out from this work is that on the one hand, biotic interactions are more important than abiotic interactions, which basically means it's more important who you live with than where you live, okay? That was one important conclusion that we were kind of surprised to see. And then the other conclusion, if we prize apart the positive associations, the organisms that live with each other, and compare them against organisms that never live with each other, the sort of positive and the negative associations, we see quite strikingly that the number of positive associations are more than 3 times higher than the negatives, sort of telling us that cooperation appears to be more important than competition in these ecosystems, which was quite a surprise to us at that when we caught this signal. We always, think of evolution as being competition, survival of the fittest. Who is strongest outcompetes the others. But in fact, no. The message of the plankton world is more telling us that it's all about cooperation, working together to ensure that the ecosystem together works. That is more important than trying to screw your neighbor and so on. So that's an interesting message that we got out of that study. And then when we look at these interactions, we look at these communities. I mean, this is complex, but don't worry. But when we look at the communities of these different kinds of plankton, and we see what determines who is present together in a particular environment, we see that it's very much temperature that is the main environmental driver determining what the community structure looks like. So basically what that means, if you give me a sample of seawater and I look at the community of organisms that is present in there, I can tell you with pretty good degree of certainty the temperature of that seawater, okay? So it's a very sophisticated thermometer that we have found here. But that's not really the point. The point is that these organisms, even the microscopic organisms, are exquisitely sensitive to temperature, which brings us back to the issue of today's ocean being very much under pressure by these increased temperatures that are happening as a consequence of global warming. So this really leads us to realize that we really have to understand how these organisms are going to change, are going to migrate, or going to adapt as a consequence of changing temperatures in the ocean in future years. And we have quite some information about that thanks to the work of the Intergovernmental Panel on Climate Change, the IPCC, who have done a lot of work bringing a lot of data together to generate models about how the future Earth is going to look in the years to come. And what I'm going to show you here is a video that begins in 1950, showing how temperature has changed on the surface of the Earth up until today, and how it's projected to change up until the year 2100. And this is pretty conservative model. Okay, it's based on pretty reasonable emissions of carbon dioxide in years to come. And what you're going to see is changes in temperature, either revealed by warmer colors showing increase in temperatures, or bluer, cooler colors as cooling events. So let's see if that starts. So here we go. So you see this moving. We're in 1960. You see it's going pretty quickly. And these are sort of temperature anomalies as some regions get a bit redder or a bit bluer. You know, everything is fairly, you know, moving around fairly insignificantly up until, you know, now we're getting through to where we are today. And we can see how much the Arctic is really suffering from increased temperatures. Here we're going into the future, so this is a model simulation based on the best available science today. And you can see how the Arctic in particular is affected. We can see the Tibetan Plateau loses its glaciers fairly soon. And by the end of the century, things are not looking very good at all, right? So this really raises the importance of trying to raise the alarm bells, if you like. You know, our signal from the plankton world about their sensitivity to temperature really made us realize that something that we really have to get the message across from the science about the state that we're actually in. So we used the climate convention, the climate conference in Paris in 2015, as a moment to mobilize the troops, to bring everybody together and create what we call the Ocean Climate Platform. All right, that was created in 2015 and continues to exist today. And this brings together all kinds of different players with interests in the ocean. Some of these you will recognize, like Scripps, the Smithsonian, the Pew Trust, various others from the US. This is very much an international operation. And the objective here was the idea was to forget about the individual interests of the different organizations, the different NGOs who may be interested in plastic pollution or extension of marine protected areas or whatever. The idea was basically to forget about the individual interests of each of these groups and just come together and promote one single voice at the climate conference in Paris, which was simply to give a voice to the ocean, very, very simply. And we managed to do that. We organized an Ocean Day during the climate conference in Paris. We brought together a lot of very important people. Here you see, that's Irina Bokova from UNESCO. That's Prince Albert of Monaco. We're all talking the same voice of giving the oceans a voice. Sylvia Earle was with us, Al Gore was with us, Tara came into Paris, Ban Ki-moon came to visit us, all together to give the ocean a voice. Incredibly, we succeeded for the first time. We succeeded to get the oceans mentioned in the climate convention, the climate agreement that was signed in Paris last year. It's a very minor mention. It's just in the preambule, okay? But you'll see here, this is what is mentioned, noting the importance of ensuring the integrity of all ecosystems, including oceans, and the protection of biodiversity, recognized by some cultures as Mother Earth. So it took this is COP21, so it took 21 years to get the ocean into the agenda, into the climate agenda. It's incredible to realize that it took so long to give the oceans a voice at this high political level. But we're very happy that working together, you know, sort of getting inspiration from how the plankton were cooperating rather than competing, that we managed collectively to raise the voice of the ocean and make it part of the climate convention. So this is really just the start of a journey. I tried to show you some of the science that is being generated from the Terra Oceans project that is continuing fast forward very much. I showed you how we're able to try to influence the political, the regulatory situation through the science, through the voice of science. We try to transmit the science that we're doing through cultural initiatives, through artists in residence that we have on board that produce different kinds of works of art, that talk about plankton, that talk about the oceans and so on. And then moving forward in the future, we expect that the science will lead us through to an increased understanding of how the ocean works, to improve our models of what the ocean is going to look like in the future, to predict our predictive power, and how to respond to the changes that are actually underway in this moment, either through intervention, through remediation, or through prevention, if we're lucky enough to be able to do that. And then finally, there are all kinds of ways in which this data, this knowledge, can be exploited through biodiscovery, through bioengineering, discovery of new kinds of genes with new kinds of functions for biomedical, industrial applications, and so on. So this is a summary as we move forward. So let me end there, and let me thank the people behind this project. It's been very much a collective effort. A lot of people, our sponsors, who have provided the money for the expedition, and research agencies for the science. We're about 20 partners, scientific partners in the project. MIT is part of it, NASA is part of it in the US, University of Arizona. And so thank you to them for their commitment to this project, and thank you to you for your attention.